Yes, good evening everyone. My name is Darshan Kumar. I'm a research scholar studying in the Department of Studies and Research in Zoology in the University College of Science under Tumkur University, Tumkuru. I am currently doing a Doctor of Philosophy in Zoology under the guidance of Dr. Parimala B, who is working as an Associate Professor in the Department of Zoology in the University College of Science, Tumkur University, Tumkuru. So now I am going to give a presentation on my research topic, that is the assessment of subchronic behavioral toxicity of Halosulfuron Methyl Herbicide in Cyprinus Carpio lineus. Halosulfuron methyl is globally it is used as a sulfonyl urea herbicide. Sulfonyl urea herbicide, it is a group of pesticide which will be having a sulfur content and urea content. This will be belongs to a sulfonyl urea herbicide. It is extensively used in the agriculture, especially in the case of rice as well as in the sugarcane. It is extensively used in Karnataka in different several district like Mandya and Shumoga and Sagra and all those different districts, and it is extensively used because it is having high efficacy at low doses, and herbicide runoffs from treated fields can pose a risk to aquatic environments. As we know that aquatic environments are very sensitive areas where the fish will survive. If any of the toxic chemicals or herbicide, it will be even go by runoff, it causes several different types of ecological imbalances to the aquaculture and aquatic fields. So aquatic organisms, especially fish, are very vulnerable to pesticide exposure and serve as a critical bioindicators of environmental health, as we all know that. So these residues can cause, that is that my chemical is halosulfuron methyl chemicals that cause several neurotoxicological and physiological disturbances, and additionally genotoxicological disturbances, hematological disturbances, as well as neuroendocrinological disturbances are observed in the fish. When we observe a neuronal axis, we will observe different types of pathological condition is observed. So there is a growing evidence that selective herbicide like halosulfuron methyl can cause toxicological and behavioral changes in freshwater species. So this is the several methodologies which I was gone that is gone through. The halosulfuron methyl was tested for subchronic toxicity on common carp, which is a test animal as a test species of fish called common carp, which will be called as an Cyprinus carpiolineus. So I taken 1 by 8 of LC50 at 69.625 mg per liter for over 40 days. So I exposed, that is a subchronic exposure was done. Already the LC50 was taken, and on the basis of LC50 I taken 1 by 8 of LC50, that is 69.625 mg per liter. So this methodology was given by Ailon and Gracia Vasquez in the year 2000. So I followed the criterias and standard procedure and protocols according to this research paper. So in a test group, I taken the 10 fish per group with 15 to 20 gram weight were taken, where one will be kept as a control and which will be another will be kept as a treated, which will be about fishes were divided into control and treatment groups with three replicates for behavioral observations was made about 40 days. So daily video tracking was done and quantified. It will be quantified neatly for locomotor activities and any swimming patterns like any irregular swimming or burst swimming. All those things was calculated, and avoidance responses and surfacing frequency. Surfacing frequency means if there is low oxygen demands which will be present in a fish species, that will be quantified, will be surfacing frequency, and feeding latency was also observed during this 40 days. The observation was made according to the actor and the co-workers. They given some protocols. According to their protocols, I made several observations, and by several tracking softwares was observed by via video trackings. So responses showed that there is a dose and time dependent changes will be observed. That means if we go for 40 days, which will be chronic observations, I was seen that is responses showed that there are several hematological parameters changes, as well as some of the endocrine level disruptions was also observed, as well as physical as well as biologically the species differences was clearly observed. There is a growing corresponding evidences was also observed, which will be observed, which will be I explain in a further slides. So as we know that ACHE inhibition means acetylcholine esterase activity was inhibited in the brain tissue, was measured bi-weekly by using several elements and calorimetric methods. These two methods was protocols was explained by Abedi and the co-workers in the year 2009. So I taken these two parameters, which will be acetyl esterase activity was observed. ACHE inhibition, that is acetylcholine esterase inhibition, was progression was observed, was 25% at day 10, where the activity means there is ACHE inhibition, that is acetylcholine esterase activity was declining, that is inhibition was progressed from 25% was observed at the day 10 and 55% at the day 40. These two, what I told, that 25% at the day 10 and 55% at day 40, this will be observed within that of control groups to that of exposed chemical group. Next, fishes were divided into mainly the control and treatment group, as what I told earlier. These groups with three replicates for behavioral observations was done. Next, antioxidant defenses was peaked mid exposure, then that will be going to be depleted. I will show in graphs. So blood parameters such as red blood corpuscles, white blood corpuscles, and hemoglobin content was estimated, was analyzed from caudal vein samples for every 14 days to detect stress responses. I recorded several hematological parameters, as well as there is a decline in WBC, there is a decline in RBC, and that will be lead to hypoxia, which means there is a several changes in red blood corpuscle cells was also observed. So my key finding is surfacing frequency was increased. Surfacing frequency means the fish will come to a surface and it will be shown that is a lateral like body was observed at the day 32, that will be causing a signaling respiratory distress was observed, and neurotoxicity correlated behavioral abnormalities was observed, highlighting that halosulfuron methyl was a disruptive potential at environmental relevant concentration. This what the key findings was I observed. So these are all the several results what I shown in the different slides. There is a behavioral parameters over 40 days. There will be observed the parameters day 10, day 20, day 32, day 40. The day parameters like locomotor activity was minus 25% was observed, and that was last 40 day 40, this completely 60% was very less compared to that of control, and erratic burst was observed. That will be wait, the one single arrow mark that shows that there is an increase, double arrow mark shows that upward arrow mark shows that there is severely increase, and triple arrow mark shows that that is the extremely severely increase in the burst. That will be, this is a comparative parameter that refers to day 10, day 20, day 32, day 40 with reference to locomotor activity, erratic burst, avoidance response, surfacing frequency, feeding rate, and growth rates. So this is what the acetylcholine stress activity and oxidative stress in controlling. That means the 10 fishes were kept to a control. That will be explained that in the days of 10, 20, 32, 40 and days, that is acetylcholine stress activity, as well as SOD activity, superoxide dismutase activity, and CAT activity means catalase activity was notified. And these are very crucial for antioxidant enzymes. These three, what is ACHE, SOD, and CAT, are very crucial for antioxidant enzyme that protects cell from different damaging, any damage signalings was observed, any damaging cells of signaling, like any a damage signal that will be coming from molecular level, that will be activated from these ACHE and SOD and CAT activities by neutralizing harmful reactive oxygen like superoxide radicals and hydrogen peroxide. These three was mainly observed. These mainly SOD and CAT are important for cellular protections and antioxidant defensives. So what I shown here, that is ACHE and oxidative stress, and halosulfuron methyl, that will be toxic chemical what I induced called as into a treated group. It shows that ACHE inhibition versus control was taken, that in the day 10 was 25%, day 20 was 35 to 40%, and day 32 was 45 to 50%, and day 40 was 55% was depleted. So this is the subchronic exposure. That means a chronic exposure, what it shows, that means there is a in a control, what green color maintained control, and red color shows treated, and here SOD and CAT antioxidant enzyme and ACHE inhibitors versus locomotor. This is what I was shown. That red color shown is a control, and what are all those, the locomotor percent activity control and all those things. So there will be a 60% locomotor activity reduction, erratic swimming bursts, from day 10 to highlighted avoidance of response, surfacing frequency increased from day 32, delayed feeding latency, impaired growth rate, dose and time dependent progression, no mortality was observed. Then ACHE inhibition was observed, that is R is equal to 0.89, which will be having a high level significance. Respiratory distress indicator, motor coordination dysfunction, appetite suppression was observed. So this is a 5-second video which will be showing that is a reverse swimming, that is backward swimming, and that is loss of equilibrium, and upward swimming also observed. With swimming activity, that is swimming was observed. This is the different types of what chemical studies was observed, the behavioral responses. So this shows that in the conclusion that there is a 55% of acetylcholinesterase activity inhibition directly correlates with 60% locomotor decline, that is R square, confirming that neurodisruptive mechanism was observed. Antioxidant exertion of SOD, that is superoxide dismutase, and CAT, that is catalase depletion, was day 40, proves oxidative stress overload. This finding established halosulfuron methyl as a significant threat to carp fisheries, demanding immediate migration for sustainable aquaculture. These are all my references. Thank you.